Hello, everybody. Thanks a lot for coming. It's a real pleasure to introduce our guest speaker to the CDCR seminar series today, Dr. David Uehling, coming from the Ontario Institute of Cancer Research. Dr. Uehling is a close colleague to a lot of researchers here at McMaster, thanks to not just a great working style and relationship, but also incredible medicinal chemistry expertise. Specializing in the science that is responsible for how to build, select, identify, design, and discover and develop drugs. So it's a real pleasure to have him here today. He is currently the interim scientific lead of drug discovery at the OICR. He actually comes to us from the US, where he did his undergraduate degree at UC San Diego. What's the name of the sports team at UC San Diego?
Tritons.
Tritons, right. The Spartans are...
More famous.
Right. He received so he did chemistry at UCSD, beautiful university. He went on to do a PhD in synthetic organic chemistry at UC Berkeley, and then a postdoctoral fellow at Yale and Harvard universities. He actually did his postdoc or a PhD with Stuart Schreiber, so I think consider the father of chemical biology as we know it. So that's super exciting. He spent a lot of time working at GlaxoSmithKline as well as a medicinal chemist. And has made a lot of tremendous contributions to the drug development field in oncology. So without further ado, it's my pleasure to introduce Dr. Uehling.
So first of all, I wanted to thank the organizers. Sheila had actually invited me, and I understand she's in Japan right now. But I wanted to thank everybody else who put together this schedule. Big shout out to Dana, and I had a great time. It was a truncated day. I came over, had some meetings. Actually, my first meeting of the day was with Anthony by Zoom, and then so the day got started on a good note. Let's hopefully it finishes off on a good note. But my day is kind of an Anthony sandwich here. But yeah, even better. That's right. So anyway, and big shout out though to Dana for all her work in putting this together, and for getting through the AV issues, which seem to be more or less solved. So anyway, I came up with the title, The Team Sport of Drug Discovery, because I didn't really know what I was going to talk about when I put together this title. But I think as I worked through the lecture, it really it started to hit home. And so I'm going to kind of expand on that theme as we go along. But my talk is consisting of different parts. I'm going to give you sort of an overview and an advertisement, if you will, for OICR and for our group. And some of you are more familiar with OICR than others, but I'll run through with you and show you how we fit in, or at least how we like to think of ourselves as fitting in. And then I want to talk more specifically about some of the type of science we do, and specifically around different drug modalities. And most of you have heard of PROTACs and how important they are, but I want to give you two case studies really that we've done in our group on how we've started to utilize this science in terms of prosecuting targets of interest before I wrap up. So OICR is, apologies for those of you who've seen this slide probably multiple times, but OICR is really comprised of the research of three different arms. There's the clinical translation, there's adaptive oncology, and then where we fit in, and my group fits in, is called therapeutic innovation. So clinical translation is really self-explanatory. It's how to move and impact the needle in the clinic. Adaptive oncology is really developing systems and knowledge to be able to understand how to better serve cancer patients. And then, as I said, sitting in the middle is therapeutic innovation, where we're actually looking to discover new drugs. And so our OICR is really, as I look at this slide, I start to think maybe it's a little bit pretentious to say OICR is the center for Ontario's ecosystem for cancer discovery and innovation. When after today and seeing the wonderful work that's gone on here, there's obviously multiple centers that do great work. And I think what the point of this slide really is that we're a very collaborative institute, and we're extremely thrilled to be able to collaborate with researchers here, but throughout the province. And so, we're kind of put into place as an agnostic group. We're a so-called honest broker who can work with different groups and not be biased towards one research institution or another, but really to be able to serve the needs of all of Ontario in terms of translating research. And so Therapeutic Innovation is the group that I represent. And our mission is to discover and to develop novel validated cancer targets. And we take them from academic researchers as well as from our own program. DD stands for drug discovery. And we discover and advance first-in-class or best-in-class compounds for preclinical development. And we ensure that these drug assets have a competitive product profile. So we have to have high standards, that we want to actually do things that are clinically impactful. So in order to do that, you have to start with a drug that really meets a lot of criteria. And I'll touch on that later on. We can't do it all. We have to actually attract further investment. So we're really focused on the early stage, and I'll explain that more in a minute. So we consist of 3 programs: the drug discovery program, and we also have a collaborative research resources, which basically is saying that we're open for business, to quote our premier, in terms of if you want to have an enabling piece of research done, say a pharmacokinetic study or a compound made, or some other thing that we can do, or even assay development work, we can help you there. And then we have the Cancer Therapeutics Innovation Pipeline, which is basically a funding mechanism, and then the drug discovery community. And so to explain a little bit more about the Cancer Therapeutics Innovation Pipeline, and I know Anthony knows about this really well. It's a funding mechanism sponsored by OICR for early stage research. And there's multiple stages of early stage research. There's early validation, which is basically you have a biological target. You don't know how important it is, but there's work to be done in terms of CRISPR knockout or other exploratory studies to help validate that this is actually a target that you really want to go after. Then there's the early accelerator stage, where once you actually have that initial validation and that comfort, then you can set up biological assays and screens. And then there's the late accelerator, which was you're actually prosecuting hits from the screens. And then finally, there's the lead generation, where you're actually developing a lead that has in vivo activity. And that's really where the whole cycle stops. And our portfolio consists of both small molecules and large molecules. And I want to emphasize that because this is open to all of the researchers in Ontario, this isn't all done by our group. And in fact, we don't do any large molecule work in our group. So you see the biologics that are in this screen in the different stages all come from academic PIs, and the work is done based on their research, even though we may be able to contribute in different ways. So it's a pretty robust pipeline, and we're looking forward to moving it in some of these compounds into the clinic through partners. We also have the drug discovery community, and we actually had one here. I think it was about a year ago, maybe a little bit less, maybe back in the fall. But it's really where we go out to different well-known research institutes throughout Ontario, and we try to build relationships. And we do it through, it's a two-way street. We talk about what we're doing, as well as we hear what different professors are doing, and we see if there's opportunities to collaborate, either informally or through some kind of a grant or funding mechanism. And I think one of the really important aspects of all this that we really are just starting to emphasize more is training and networking. And I think there's really opportunities for people in places like this to come to our group and actually get trained. And we have, I think, a way of doing things that would help train people for industrial or pharmaceutical research or academic research, depending on which way you go. And of course, we want to be able to engage with the industry. And we have a commercialization group called Facet, which is helps fund, and they actually help do the licensing of our assets. So This is just a chevron. Many of you have seen this sort of drug discovery continuum series of chevrons where you starting from the earliest stage of target validation, and then the ultimate stage of a drug discovery project is review and approval from a regulatory agency before it actually reaches the market. And so this is one of the things that is often confused with our group is we don't do drug development, we do drug discovery. And it's a very distinct difference between the two activities. Drug discovery is where you're actually setting up and you're actually identifying a potential clinical candidate. Drug development is where you're taking that clinical candidate and going all the way into the market. And, as I mentioned about this funding, we stop at the point where we're doing lead generation, where we have compounds that actually have in vivo activity. But really, what we are seeking to be able to do through partners and additional funding mechanisms is go into lead optimization, where we actually discover the clinical candidate. One of the projects I've been on, we kind of managed to make it farther than our remit, if you will, and discover a clinical candidate. But typically, we're supposed to partner at that stage where we've just discovered an in vivo active compound. And the whole idea behind all this is that we want to retain and increase the value of Ontario developed IP to the greatest extent possible before partnership with a third party such as a pharmaceutical company. So we're here to try to add value to any project that comes out of an academic environment. And so this is some of the faces of a drug discovery group. We have, myself, you heard about. Thank you, Anthony, for that very nice introduction. Actually, I'm stepping in for Dr. Rima Alawar. And Rima was actually the one who was instrumental in building up the group from the ground up some 16 years ago when I joined the group. It was a small team. We had a bolus of medicinal chemists come in. But then we realized that it's really a naive to actually have just medicinal chemistry, which was kind of the focus of our group. We actually have now biology and other functionalities. So we have Richard, who's leading our biology group, Masood, who's actually protein production and X-ray crystallography, and I'll share some of that work soon. Jay Ramnath, who's kind of the glue that holds our group together. He's the project planner. Laurent is our computational chemist, and Meth and Isaac, who some of you know well, and Ahmed Aman, they're medicinal chemists and people who do analytical chemistry and pharmacokinetics, respectively. So I think we have a team that actually has a lot of industrial experience, because most of the people here were hired from industry as opposed from academics. And I think it gives us a little bit of a different mindset to help mesh with and interact with the academic community. So this is just showing that getting back to the teamwork aspect of things, we have a group that has a lot of different expertise and functionality. The yellow boxes there, I think they're yellow. Yeah, yellow or orange is really related to chemistry. We got computational chemistry. We have compound libraries, medicinal chemistry is kind of the core. And then we have biology in terms of compound screening and assay development and protein production. And then we have some of the more elaborate studies. We're actually developing the drug for pharmacokinetics and evaluating in vivo activity. So all these things come together, and what we're really trying to do is bridge the gap between academia and industry. So just to give a little bit of a highlight for some of our emerging platforms or technologies, and I won't spend much time at that. It's just basically to try to pique your interest. We have a computational chemistry group that's actually developing some machine learning approaches to drug discovery, and they developed a platform called QSL, which is really a joint venture between our group and CogniFrame, which is a company that specializes in delivering quantum computing. And so they're looking at different stages of solutions for early stage drug discovery, but it's really taking a look at leveraging quantum simulated chemistry to actually optimize compounds. And there's multiple aspects to this. There's where you actually can do virtual screening to find compounds that first bind to a protein and actually figure out what pocket it binds to begin with. Then there's an aspect to it where you can actually have the computer design drugs for you, and we're starting to leverage that more in our group. And then there's QBind, which is basically a predictive tool to assess how well a compound will bind into a known pocket. And so we're looking at this and working with the academic community, different partners, and of course with our group as well. And then one other emerging platform I just wanted to highlight was our lipidomics, and that's really being spearheaded by Ahmed Aman, who was the last guy I showed in that picture, in that set of pictures. And he's developing, in his group, a system where you can actually look at lipid profiles of different cancer cells and be able to do biomarker discovery, target discovery, and drug discovery, all to be able to impact things. And he's gotten some really exciting collaborations with some of the people in the Toronto community to actually characterize different subtypes of cancer, but also look at how cancer cells change with different markers. And I think it's going to be a very versatile platform that we're going to be using both in our own programs as well as working with clinicians. So switching gears a little bit, I just wanted to highlight that we have compound libraries that we're willing to share. And this is just showing that we have different sets of compounds. Talking with Hong earlier about different types of compounds to screen, but you can see here's kind of the whole gamut of compounds, ranging from huge compounds that have no pre-assigned characterization. We don't know anything about these molecules other than the structure and their computational properties, versus more focused sets where there's a lot more known about the biology of those different targets. And then there's compound sets that are designed based on their particular structural features, and they're curated so that they're more likely to hit different classes of targets. And so, the point here I want to make is just that actually, not only are these some of these libraries housed in McMaster, quite a few of them actually, but we're able to share and do any follow-up work in collaboration with the academic institutions, be it whether they need solid powders and to confirmation of the hit, or actually even doing SAR, structure activity relationship medicinal chemistry, around the different hits. And so this is just something that's we like to advertise for all of our potential collaborators. And of course, I wanted to highlight McMaster. It's been we've had a great partnership there, and we've actually had a lot of screening, and I'm sure that these libraries can be used in the future here as well. So now just getting more into some of what we're doing in our group science-wise. And I just wanted to highlight the fact that we're really a small molecules group. That's our focus, and we don't do proteins. We produce proteins, but not as drug such as antibodies. We're really focused on the small molecule medicinal chemistry aspect of things, but we want to be able to leverage that capability in different modalities. And so the three that I'm highlighting here are induced proximity and protein degradation. And we have projects that I'm going to be telling you about. PROTACs, we're interested in molecular glues, and we have one early stage project there. And of course, we have the CAM project with Anthony that I'm not going to be telling you about today, but it's just one of the examples of the concept of induced proximity in a kind of a grand way. And we're also have efforts in antibody drug conjugates, not the protein itself, but we work with the NRC on and actually linking our chemistry to antibodies. And last, we're actually now just recently getting into radiotheranostics, and we're working closely with the Center for Pro-Development Commercialization. But a big shout out for the fact that these collaborations and some of the other ones that I'm not even going to tell you about are a lot of them are with McMaster, and I think this is just because of the environment here. I think it really is well suited to synergy with some of these emerging areas. So what I'm going to be focusing on for pretty much the remainder of my talk is what we're doing in the induced proximity and protein degradation area. And I want to just tell you about a little bit of a background, which most of you probably, some of you may know more about than others, but really chemical induced proximity involves when you have usually small molecules. So it doesn't necessarily have to be two small molecules to bring two proteins into spatial proximity. And when you have that proximity inducing effect, then that actually often causes an altered function of the target. And this is a very, very attractive approach for targeting drugs that are proteins that are considered undruggable. They may not be enzymes, they may not necessarily have a pocket that has a function, but if you can target these pockets, you can actually cause, through induced proximity, different things to happen. And there's a lot of different aspects to this, and a lot of different companies are actually cropping up to which are exploring this different area. That's really the sky's the limit in this particular area. Now, a subset of this is called proteolase targeting chimeras, or PROTACs for short. And it's really the concept is based around, as this cartoon shows, hijacking the ubiquitin proteasome system to affect protein degradation. So in essence, what happens is you have a protein of interest and you have a molecule which causes induced proximity between that protein of interest, generally one that you're trying to degrade or downregulate, and an E3 ligase, which then ubiquitinates the protein and causes it to ultimately degrade. And so these E3 ligases are there's an expanding repertoire of E3 ligases, but the most common by far are cereblon and von Hippel-Lindau, VHL for short. And these are the ones that have been used the most commonly. And so I'll refer to some of those as we go along. But the whole idea is you're targeting a protein of interest through this new way of harnessing or hijacking the ubiquitin ligase system. And so there's a number of PROTACs that are already in the clinic, and we've gotten as far, I think, as phase 3. There's a company called Arvinas, which is really kind of the leader in this space, and they have a number of different targets for different cancer-related proteins. But you can see this is a nice representation taken from this review of all the different target proteins that have been looked at. You have BCL, BRD9, which is actually kind of the poster child because it's so easy to degrade, EGFR, BTK, and a number of these other ones. And these are, generally speaking, almost all of these are closely related to cancer, or they're well-known oncology targets. And then you have the E3 ligases, and then you have various ones, VHL, cereblon, and other ones that people are working on. And I'm going to tell you about some of our novel work in that area. So, so the first case study or example I wanted to tell you about was around HPK1. And HPK1 is a well-known immuno-oncology target. And this is a fairly complicated graph. I wish I had a pointer, but you can see the blue circle there is circling around HPK1. And essentially, for simplistic way of looking at it, is it's in the T cell, and it actually is a down regulator of immune stimulation. So it actually works by causing phosphorylation of its primary target, which is SLP76, which you can see in that red circle. And SLP76 is really the immune mediator. It's one of the master immune mediators, signaling in terms of signaling transduction in the T cell. And it causes all the things to happen in the T cell, which ultimately result in T cell proliferation and a release of cytokines. And so if you inhibit HPK1, you're essentially inhibiting the inhibitor of that system and causing immune stimulation. At least that was the theory that was caused. And so HPK1 should have a potential, if you can develop a selective inhibitor, to be either working alone or in combination with standard immune checkpoint inhibitors and that are well known in the clinic. And so you have had the situation where HPK1 inhibitor should stimulate a neoantigen through NK activation. It should increase cytolytic activity of cytotoxic T cells, and that's really the primary aspect that we're focused on in this project. And then there's effects on other cells in the immune compartment. So, if we have an inhibitor, it should do, all the things that we're hoping to do, which is to, first of all, have the direct mechanistic effect of inhibiting SLP76 phosphorylation, and it should increase the release of cytokines, and also should have specificity of that kinase. We need specificity because if you're inhibiting off-target kinases, you can actually have the opposite effect of inhibiting HPK1, and that's going to basically shoot yourself in the foot. And so in any drug development or drug discovery campaign, you really have to figure out how to get as much selectivity as possible so you can reduce the side effects. And HPK1 is a very interesting protein. It's a kinase. Kinases, they make a living by phosphorylating other proteins within the cell and or on the cell membrane. And the structure of HPK1 is shown here, and it has an interesting domain swapped interface. And we actually had structures solved by multiple partners. We had one done from outsourcing, actually, from a company in Germany called Proteris, which did a great job. But we also have a long-standing collaboration with Gil Privé at UHN. And both teams essentially solved the structures using slightly different techniques. The Proteris team actually used mutations to actually cause a cysteine binding or a disulfide bond, which actually created this domain-swapped dimeric interface, whereas Gil Privé solved the structure in the native form, if you will. And in each of these structures, you can see that there's two molecules that are bound to HPK1 because it is this homodimer, and those are represented in the black squares. So one of the key points I want to make here is that HPK1 is actually a member of a family of kinases, MAP4 kinase. There's multiple subfamily members, and one of them, GLK, is actually, if you inhibit GLK, you have the opposite effect of inhibiting HPK, and yet the structure is almost identical. It only differs in the active site, the area that you're looking at, by only a few amino acid residues. And so going into this, we were very concerned about how well we could actually get selectivity from HPK1 versus GLK. And so, in order to start this project, we used one of the compound sets that I told you about earlier called the OICR kinase inhibitor library. And just a little bit of background, back in 2009, when I joined the group, we'd actually started this campaign to get a set of compounds that are known kinase inhibitors. So these are not proprietary compounds. They're compounds that either you can get from Celic or you can get from patent. A lot of them we had made. And there's compounds that are compounds that are even from the primary literature, like a JMed Chem paper that might explain here's a new inhibitor of a kinase. So it's actually quite a diverse set of compounds. But the good thing is that these compounds may not be specific, but we know at least one kinase target for all of these drugs. And we're up to, actually, this is a little bit out of date, we're over 800 compounds now. I think we have 880 compounds, but it's been a very useful library. And we've actually teamed up with academic partners to do cell-based phenotypic screening. We've identified hits for novel targets using that approach, and we probe new biological hypotheses, because if you have multiple compounds, and this is another point I want to make, is that there's kinases that are inhibited by multiple drugs, and so you have that level of redundancy that really gives you a higher confidence that if you're if you have multiple compounds doing the same thing and they all hit the same kinase, then you feel better that that's actually the true target. But we've also applied this, as I'm going to say, to internal projects. And so we can either probe biological hypotheses, we can explore drug combinations, which we've started to do, or we can identify hits for novel targets. And that's exactly what we were doing here. And so we started to, and apologies, I can't show all the structures yet because they're still under patent. This is one of the frustrating things about this, but in any event, I'll be happy to tell you a little bit more about the chemotypes at some later point. But we actually started our lead campaign by screening at the kinase inhibitor library at 300 nanomolar, and then we did IC50 follow-ups, and we found this well-known drug called crizotinib, which is actually a billion-dollar drug targeting a subset of lung cancer patients. And its primary target is ALK, but it also inhibited HPK1. We were trying to get rid of a target called LCK because that was one of the kinases I was telling you about that does the opposite of HPK1. So if you inhibit both HPK1 and LCK, you're not going to get a good phenotype. And so you got to figure out how to get rid of LCK. But we did some more medicinal chemistry. We got from crizotinib to this compound called 14779, that's our code name for it, that started to work a little bit better in cells. And lo and behold, it actually had better selectivity on LCK against LCK as well. And so then we did a lot more medicinal chemistry. We made up 2,000 compounds, over 2,000 compounds. And we then got to a point where we got compounds that had really good LCK selectivity, good PK, pharmacokinetics, a lot of the other properties that we were seeking, but we also had poor GLK selectivity. And finally, with some more medicinal chemistry optimization, and I would like to say that we did de novo design to actually get this selectivity, but it was really basically around where are the amino acids different between GLK and HPK1. And then we started to make changes around that were educated guesses, I would say, to get better selectivity. And we finally came up with our candidate compound 35700, which meets pretty much all the criteria. And just to show you what this drug actually does in vitro, and these are human primary T cells, so they're harvested from the blood. And what happens is you stimulate with an antibody, CD3, CD28. Anti-CD3, CD28 causes sort of a basal stimulation of these cells. But then when you treat them with an HPK1 inhibitor, what you see is as you're increasing the concentration of the compound, you see a much higher stimulation of interleukin-2, which is one of the key cytokines from T cells, as well as interferon gamma. And interleukin-2 is always the most strongly stimulated relative to interferon gamma, but we think they're both very important cytokines to affect an immune response. And we also see a slight increase in proliferation. That's those blue dots. And the green triangles basically indicate that this compound is actually working on mechanism because you're inhibiting the phosphorylation of SLP76, which is the primary target in cells of HPK1. So you want to know that these effects are not due to some off-target effect, off-target kinase or something else going on. And that's one of the things that I think we're try to be as rigorous as possible about, is actually really lining up the dots to make sure that our drugs are working the way that we think they're working. And so, one of the things that was encouraging, and what this blue square really represents, is the fact that earlier on in our program, before we had good selectivity, we had these sharp sort of spiky curves, because as you start to inhibit HPK1, then you're going to start to inhibit other kinases, which cause a decreased effect. And we still see with 35700 that bell-shaped curve, but it's over several log orders of concentration. So we think that kind of the area under the curve is really what's important. So then going into an in vivo setting, we use this model called the EMT6 syngeneic model. And what we actually saw was the control animals are represented by the black dots. And this is a model where you're actually implanting a mouse tumor into a mouse with an intact immune system. You have to use mice with intact immune systems, of course, because you're actually taking advantage of the immune system to cause your anti-tumor effect. And so the black dots represent the size of the tumors at the end of the experiment for the non-treated animals. The green dots represent the size of the tumors for animals that were treated with a checkpoint inhibitor, an anti-PD-1 inhibitor, which is kind of the standard of care, if you will, for the mouse here. And then the red dots are the combination between our drug and PD-1. And so, it's natural then to ask, well, what does our drug do alone? And if you look in this graph, it's a different way of representing anti-tumor effect. And this is probably a more common way. You're actually looking at the tumor growth over time. And the black triangles represent, the black circles represent the size of the tumor as you go through the 21-day experiment. And the green and the red are our compound and anti-PD-1 as single agents. And then the combination is the blue triangles. And one of the things that we wanted to do is, we wanted to make sure that we were actually getting this effect not from a primary anti-tumor effect, that is killing the cell directly, cancer cells directly, but we were causing the immune system to kill the cancer cells. And so we did this experiment where we knocked out the immune system of the mouse. And as we expected, when you knock out the immune system, the controlled and the treated animals behave more or less the same way. So that was sort of a more definitive experiment to show that we were actually working through this mechanism of action. So this is just a graph, and I'm not going to go through all these numbers, and I'm not trying to get you to digest all this. It's really just to show you that when we get to a lead compound, we do a lot of work to see how it matches up to the so-called product profile. And you can see the product profile is represented in the kind of the gray squares, where these are the parameters that we're shooting for the drug. And then when you see the green, that means we've met those criteria. And then the yellow means that we were close, but we didn't quite succeed in doing exactly what we were hoping to do. We were getting there, but one of the key parameters that we didn't quite achieve was GLK selectivity. Now, how important it is to get 50 or 100-fold, we don't really know, but we know that the more selective, the better. And we were shooting for at least 50-fold, preferably 100-fold. And the best we could do here was around 30 to 40-fold, and in some cases with our lead compound, even less. So that's where we also were taking a different approach, and that's coming back to the PROTACs. And there's a number of rationale for pursuing HPK1 PROTACs in general, and that is that there's been publications shown that HPK1, as I said, is a kinase. It phosphorylates another protein. However, it's also thought to have scaffolding properties. And so whenever somebody is developing a PROTAC approach, one of the first things to an enzyme, the first thing that one asks is, well, why do you want a PROTAC to degrade the protein completely? Why don't you just inhibit it? But the theory was that, and some of the literature suggests that HPK1 is more than just an enzyme. It actually has scaffolding functions, which are important in the immune system. And so, we had this proprietary series of inhibitors. And so what we thought to do is just create PROTACs out of them. And the idea here is represented in this graph, as you can see in the middle, where you have a PROTAC has a part that's targeting the HPK1, but it's also bringing in the E3 ligase, which and generally in this case is called an E3 ligase called cereblon. So the idea is we build these molecules, and then we cause degradation of HPK1, not just inhibition. And so we made one such compound. In fact, it was the first compound that we made in that area, and it was codenamed OICR 39349. And it's pretty potent on HPK1, and we were actually surprised that putting all the extra stuff, which is on the right-hand part of this molecule, where you see the HPK1 binder, you see the linker, and then you see the cereblon binder. All the linker and the cereblon binder, it doesn't seem to necessarily inhibit it, or it doesn't seem to affect HPK1 activity. In fact, it might even help it. But what you can see is that the selectivity with GLK is not that great. But we were encouraged by the fact that when you pre-incubate T cells with this molecule, you actually get a more robust T cell stimulation, suggesting that this was actually working on mechanism, that it was degrading the cell because it was working better after the PROTAC had a chance to degrade the target protein. And so we did some definitive experiments to show that taking 39349, which is our PROTAC, and actually measuring the levels of HPK1 by Western blot, you can see that there's a nice decrease in the levels of HPK1 over time, which is just what we wanted compared to the control. And interestingly, just HPK1 inhibitor alone and not the PROTAC does decrease the levels of HPK1, but not nearly to the extent as the PROTAC. And so this is just another way of representing. We developed this high throughput, or more or less high throughput assay to measure HPK1 degradation. And for those of you probably who work with Western blotting, you know that it's a very labor-intensive, difficult technique to do on a high throughput. And so with this NanoLuck assay, you're actually measuring protein degradation by tagging HPK1 to a fluorophore. You're watching the degradation in real time, and you're actually able to measure degradation really quickly. For some reason, in this assay, it doesn't go all the way down to zero, but we think that's an artifact of the assay. The point is we're able to actually compare compounds and screen them relatively quickly to make sure that they're actually having an effect. So you can see some compounds degrade HPK1 a little bit, but some compounds degrade better, but we can actually use this as a routine assay. And so, what is this all good for? Well, we wanted to compare our small molecule to our PROTAC to see whether there really was an advantage to degrading the protein as opposed to just inhibiting it. And so on the left, you see 35700, which is just a small molecule. It's not the PROTAC. And you can see that this is just another experiment in a single donor T cell showing that you're increasing interleukin-2 and you're increasing interferon, and you're also inhibiting SLP76 as you would expect. But what we see is that with the PROTAC, we get a much more robust response where you're actually stimulating IL-2 and interferon gamma much more. And more importantly, or at least equally importantly, you see this bell-shaped curve with the small molecule. And on the right, you can see over more orders of magnitude, a higher, more sustained response. So you're not seeing the belling or the drop-off as much. So we think that that's the advantage of the PROTAC. Even though it biochemically inhibits GLK in the cell, it doesn't seem to be manifesting itself in terms of that bell-shaped curve. And this effect becomes even more pronounced with another experiment we're doing. The whole idea is that cancer cells or tumors are very immunosuppressive. They're in this immunosuppressive environment, and what you're trying to do with an IL agent is trying to overcome that immunosuppression. So you can model that using prostaglandin E2 and NECA, which basically are the surrogates for what a tumor might see and what you're actually experiencing in a tumor in terms of that immunosuppressive environment. And so you can see that the responses are quite blunted, and you can actually, if you have enough of these agents, you can actually completely reverse or stop some immuno-oncology agents from working. We do see some response with our HPK1 small molecule inhibitor, which is good. But what we were encouraged about is that with this PROTAC, we see a much more robust response. And so we think that that's going to be really to our advantage. And so currently, right now, what we're doing is developing agents which have the pharmacokinetics and all the drug-like properties so that we can demonstrate this in vivo. So it's an ongoing story, but we're very encouraged by the cellular data that we've gotten so far. So now I'm going to switch gears a little bit and talk about a different protein that's been kind of near and dear to our hearts, and it's called WDR5. And this is not a kinase. It's actually a member of a family of WD repeat proteins, which has been an area of intensive investigation in our group. And WDR5 is a member of this family of proteins. And what it does is it's actually an essential gene, and it's related to oncogenic processes, and particularly in leukemias and lymphomas, but also we think in solid tumors as well. And it's actually, you can see WDR5 is a member of a very large complex of proteins, and they all work in synchrony to basically cause epigenetic changes of the histone, for example, acetylation and in this case, methylation. And the Structured Genomics Consortium, who's been a great collaborator of ours, actually saw the structure of HPK1, or sorry, of WDR5, and you can see it has a characteristic donut hole where the peptide, which is part of the MLL, actually is a subset of the MLL binds. And so what we're trying to do is block that interaction. And so we did a great collaboration with SGC to develop what's called a chemical probe, which is a molecule that has a certain level of specificity and activity on the protein, but it may not be a drug-like candidate, and it can be useful for doing biological validation studies. We took that molecule and we did some medicinal chemistry, and we got a development candidate called 12256, and that showed really good in vivo efficacy. This is sort of a different type of in vivo model. This is actually a immune compromised model where you're putting a human tumor into the mouse system, and you're seeing how the tumor grows with your compound being treated or without. And you can see the vehicle grows much faster, and the tumor, the cancer cells are working the way they are supposed to work. But so we actually started working in that family. We had a long history of that, but we thought it would be interesting to apply this target to PROTACs. And so kind of a related staggered parallel study that we did was actually to explore another WD family, a protein called DCAF. And DCAF is actually, it's called DDB1 called associated factor 1. So that's why you call it DCAF1, because it's so hard to say. But DCAF1 is actually that donut-looking protein, which is in this complex, which actually brings, and its function is to bring substrates into proximity with the ubiquitin ligase and cause degradation of the protein. And I can tell you, these are really, we like to joke because these proteins, as a general rule, look a lot like a donut, and they have a donut hole. And this looks like a cruller you can get from Tim Hortons, but hopefully not the same color. But in any event, it's got these 7 repeat blades, and we've been very interested in these type of targets. And so we've been looking at this for in terms of drug candidates. So we started off and we did actually working with a biotech company, SGC, in collaboration with us, called XChem, and we actually used a DNA encoded library to get an initial hit. A DNA encoded library is basically just a way to make a large number of compounds that are tagged by DNA and actually be able to deconvolute them very quickly. And so this is the lead compound we got, and we were able to do optimization to this chemical probe, which we then were able to use to hook on to different WDR5 binding ligands. And so we did this chemistry where we made these different PROTACs, and because this is an E3 ligase, we were hoping that by hooking onto this, by joining the E3 ligase with WDR5 through these PROTACs, we'd be able to get degradation. And sure enough, we did. You can see that we're getting some nice degradation of WDR5 through these systems. And so we're actually very excited about this. And then one of the most exciting things I think that happened was we were able to actually solve a protein crystal structure that has a ternary complex of WDR5, the PROTAC that joins the two, and DCAF. And this is the molecule that is represented in this crystal structure. So we're very excited about that. And I just wanted to finish the story by sharing with you an exciting early unpublished result, which is that getting back to the HPK1 project, we actually used these decaf handles. Here on the lower left, you can see the degradation of HPK1. And you can see that this compound 412, most of these have cereblon E3 ligase handles as their handles that co-ops E3, the E3 ligase. However, we did make one compound called 41265, which actually used a decaf binding unit. And so what we actually saw remarkably is reversal of selectivity between HPK1 and GLK, and by switching the E3 ligase. And we're not sure if this is therapeutically relevant because we're not actually trying to inhibit or get rid of the GLK, but there may be other applications outside of oncology for that. So it was a very interesting and unexpected result, but it just shows the power of being able to selectively target different proteins through this approach that you couldn't necessarily do with a small molecule. So, and just to wrap things up, I just wanted to say that we're an interdisciplinary group. We have small molecule expertise. We have oncology biology expertise, and we're here to help researchers throughout Ontario in collaborative ways. And with this particular talk, what I focus on are a couple of different projects for oncology, which rely on PROTACs, which is really an emerging and exciting field in this space. And we think that they can give us a better biological understanding of targets, but also ultimately what we're all about is trying to deliver a better therapeutic benefit. And so these projects would not be possible without multidisciplinary and interinstitutional collaboration. And that's really that's key to our success. And so, just to really highlight that point, I think in drug discovery, it's a very daunting task. I think many people work throughout their whole careers, not ever discovering a drug, but if you're not collaborating with the right group, you're like that guy there who's not going to be very successful. But having the collaborations is really what leads to the success. And so we've been very fortunate that we've had great people on our team being able to prosecute these targets. This is HPK1, some of the collaborators. I highlight some of the people in the chemistry and the biology and analytical chemistry and computational chemistry side within OICR, but also we had collaborators from UHN who have been really instrumental in driving the success of the project through their structural biology work. And then finally, we had some people from SGC, from Piramal, and from Merck, all collaboration along with these companies who are working on the decaf project that I told you about. So it's been a great ride, and it's can continue to be so. One of the things I think is McMaster and from my tour today and from everything I've known from seeing how things go here, you're a very collaborative group, much more so than I could say various other people in unnamed institutions. But I think that it's really very rewarding. And I think we're very excited about trying to figure out new ways to collaborate and to be able to expand our current collaboration. So this is just our group and some of the key players in our group. But you see Rima Alawar in the center. She just broken her ankles, both ankles simultaneously, playing soccer, and that's why she's in the boots. But anyway, it's been a great group to work with. I just want to finish up by thanking you for your attention, and again for having me spending your late Thursday afternoon with me. Thank you very much. And I think I went a little over, Tom.
That's perfect. Great. Take some questions from trainees. Yes.
Yeah. Points that were effectively here to the PD-1, targeting.
Yes.
You put those tumors back in the mice. With the immune system, you have memory to take up the tumor?
That is a fantastic question. The short answer is no. That's something that we want to do because, I think there's a couple of different experiments that these experiments, we actually do them, I didn't actually say through Wuxi. We don't do them in-house. We actually do them through a CRO. And they're kind of expensive studies. So we have to be very careful about how many we can do and what funding we have. But I think that's next on the docket, is to be able to do that type of experiment. Also, just to see how stopping dosing and seeing which tumors grow back, because the thought is that this is going to be a curative as opposed to just inhibiting growth. So many of the projects I've been involved with have been just inhibiting the growth rate of the cell, the tumors, as opposed to actually curing them. And I think some of the mice look like they're showing signs of curation, but doing the memory effect is a really important study, and it's actually been suggested to us by multiple outside people. So great question. Yes.
With regards to the what about behind paddle binder on the other side instead of just the binding itself? Is it like it's too bulky, or can you imagine it's more effective than like the bigger boards together?
So if you're saying, why can't you just have an E3 binder and the molecule separate without being attached? Is that?
No, why not? Why not have it like itself instead of having a binder that recognizes things like it's an implicitly like you're
I think you mean having like a soluble protein as part of the drug that you introduce. I don't think it would go through the cell. Does this need to go intracellular?
Yeah, this is an intracellular target. So if what you're suggesting requires getting into the cell, it might be a little bit difficult if you have a soluble protein. Yeah.
Is that what you're asking?
Yeah.
Okay. Yeah. Thank you.
Like a biologic type that would go inside.
Yeah. That's right. Yes. Great.
Naive immunology question, Kevin. So very welcome just to say I'm out of my depth. But with these HPK1 antagonists that stimulate T cells, well, first of all, are they in the market already like this, T cell stimulators? And then secondly, would they be antiviral? Would they shorten the length of a common cold, something like this?
Yeah, both excellent questions. I didn't tell you about the competition, but there's actually a lot of people that are developing HPK1 inhibitors. None of them are in the market, but there's probably, when we started into this project, we thought we were among the first. And one of the disadvantages of being in a small, relatively small group is we found ourselves competing with probably 20 or 30 big pharmaceutical companies who are all pursuing that same target. And not surprisingly, some of them gotten there ahead of us. And the farthest one along is a company called Nimbus, and they're in phase 2 now. And they've started to show some in vivo activity, but I think the real challenge for this project is figuring out what the best patient population is, how we differentiate ourselves from anti-PD-1. If we can figure out, I think there's a patient population that's really going to benefit. For whatever reason, they were seeing activity in renal cell carcinoma in particular. So whether they actually go full force into renal cell carcinoma as their primary indication, I'm referring to the Nimbus, that is not clear. But I think there's going to be a lot of activity, differential activity in different tumors. But the problem is we don't really have the clear biomarkers to figure out what those tumor types are. And I think that's a real challenge for the field. But stay tuned there. And then the second part of your question was around
Would they be antiviral? Would they
antiviral, yeah. Yeah, so stimulating the immune system. Yeah, that's actually been our former president, CEO, Laszlo, was really big into that concept. I think that's something that needs to be explored. It's a great idea. Yes, Kenny.
I just have a question a little bit more conceptually about PROTACs.
Yes.
So it's co-opting the innate E3 ligands.
Correct.
Is there any consequence then on protein degradation on targets other than your target of interest? I mean, if it's pulling it over to degrade your target of interest, do you see the same degradation of the innate targets? And kind of on the same path, do you then see cells that have an upregulation of unfolded protein response then being more sensitive or less sensitive than to these.
Yeah, that's a really great question. I mean, you know, these E3 ligases are doing something anyway.
Yeah.
They're working, they have a purpose in life. And yet we're hijacking them to do this one specific thing. And is there a negative consequence to that? Or is there even a beneficial consequence if maybe you're stimulating an unfolded protein response? I don't know the answer to that, but that's an area that's definitely worth studying. That's a great question. Yes, Hom.
See, the HPK1 expression is higher in T cells compared to like a macrophage or even some tumor cell. Is that one of the consideration why you actually developing a small molecule for certain target? You require a specific expression of that target on either immune cells or the tumor cells. And then go a little broader for that question will be what other criteria will be considered for you, whether it's a good target to develop for a small molecule inhibitor or.
Yeah, really, another great question. I mean, HPK1 is pretty much limited in expression to the immune compartment. So sometimes you think of a cancer cell in terms of, protein targets for cancer as being the primary target. So you're trying to kill a cancer cell selectively. So you're looking for proteins that are either upregulated or specifically in those cancer cells. This is actually a little bit of a different situation where you don't really care about HPK1 in the cancer cell. I mean, I don't think that there's any really meaningful benefit to inhibiting HPK1 in the cancer cell. You're specifically inhibiting it in the tumor cell. I mean, sorry, in the immune cell, so that it can kill the cancer cell. So it's not really your typical cancer biological target per se. But you're right. I mean, and typically you want to look for protein expression patterns, you look for activation, and you want to be able to see whether or not you're actually overexpressed in the tumor cell. So that's an important consideration. If you're expressing in every cell in the body, maybe you can get away with inhibiting it, but you want to know what it's going to do there in these other tissues. So it's a very important consideration, I think. And a lot of drugs have probably failed for that very reason, that they are not specific enough to the tissue of origin. And that's where actually getting to your question, this is what the whole field of antibody drug conjugates and radiopharmaceuticals, which this has been so famous for, is basically figuring out new ways to get cytotoxic agents specifically to the tumor cell. And the traditional chemotherapies basically inhibit, say, the tubulin interactors inhibit all the cells that are dividing in the body. And so if you can figure out a way to get them specifically into tumor cells and be able to bind to cancer cells over other cells, then you got a leg up. Yes.
For the longer term treatment with the K1 inhibitors, you see like more exhaustion markers, perhaps just relating through the, I guess, have native T cells in the pathway like that, like maybe 2 or 3 weeks, and also looking at different tumor microenvironment, just more things that are making T cells prone to exhaustion.
Yeah, those are great questions. I mean, I think we haven't done the experiments yet to actually show that if you continue to inhibit, how it's going to affect it. And I think you probably have some great ideas of how you would design that experiment, which we haven't gotten around to doing. We're really looking at the transient effects, but that's going to be really a relevant question over time when you're dosing in the clinic and how long it's going to actually work. What we have seen is that HPK1 inhibitors seem to reverse exhaustion markers for T cells, de novo, when you're actually, looking at the first treatment. How does it impact the T cells that are already there? But your question is a really good one, because it's getting at another question: is can you get exhausted from the agent which is causing the reversal of the exhaustion to begin with? And that's something that we really haven't explored very well. Good thought.
Rebecca.
Yes.
Because checkpoint inhibitors can have, because it's a fairly broad immune activator, there could be toxicities associated with it, but it's more anti-CTLA-4 one. But I'm just wondering if the inhibitor because it's also fairly broad, could enhance some of those toxicities, if you're just broadening that.
Yeah, I think, yeah, it's a great question too, because, you know, we don't know, we feel some reassurance that, you know, Nimbus has gone into the clinic and they've actually see what's a therapeutic window. But I was kind of interrogating the guy at the poster session from Nimbus about toxicity, and he was really saying that there was a, he was implying without saying, because he was very cautious about how he's putting this, representing the company, but he seemed to suggest that there was a narrow therapeutic window. Whether that was caused by the mechanism or their particular molecule or how they were dosing it, those are all open questions. But, you know, when you're playing with the immune system, you're playing with fire. And this is that kind of the dark side of immuno-oncology agents, all immuno-oncology agents. So how HPK1 is going to stack up to these checkpoint inhibitors remains to be seen. And maybe in some context, I mean, ultimately, if you're getting good efficacy in tumor types that you know, aren't being treated well by any other therapy, you can really tolerate that limited therapeutic window. But I think there's always going to be that issue.
Cost benefit.
Cost benefit, yes. Good question.
Well, some pretty great conversation with the audience, but I guess
Yeah.
in the interest of time,
Yeah.
let's thank Dr. Uehling for a great engaging presentation.
Thank you.